Hello, my name is Eddie Rubin. I'm the director of the Joint Genome Institute. I'd like to welcome you to the 7th annual JGI user meeting, Genomics of Energy and the Environment. Over the next 2.5 days, there's a variety of, I think, very interesting talks on genomics, energy, and environment, combinations of those words. Some will have just one, some may have all three. There are poster sessions. There will be a poster session this evening, right after the keynote talk. There'll be a poster session tomorrow. There'll be buses that'll take people from here to the JGI, and there'll be posters in the evening. There's food with the poster sessions, and there'll be tours tomorrow of the JGI. Tomorrow in the afternoon, there'll also be an interesting session which we're looking forward to everyone, as many of you participating in, and that is a session to brainstorm about how an institute like the JGI user facility evolves in the changing genomic landscape. I'm very pleased that our keynote speaker is Carl Zimmer this year. Carl is a renowned American scientific journalist. He received his undergraduate degree from Yale in English. What do you do when you have a degree in English? You look for a job, and he managed to find one at a publishing company. They happen to be publishing Discover magazine, and he got hooked on science and has, over the last period of time, has been a weekly reporter for New York Times on science, biological news, and various biological stories. He's on the frequent contributor to Discover magazine, to Scientific America. He's received numerous awards for scientific journalism, including awards from AAAS and from National Academies of Sciences. He's written a dozen books on various science topics. They include some of the topics near and dear to the hearts of many of you. It includes a book on E. coli, microcosm, a book on viruses, a book on parasites, and in fact, he even has a parasite that was named after him, a tapeworm. And he recently published a book of tattoos, scientific tattoos, and on scientists, and there are explanations for them. And I think it is really timely to have a science journalist speak to us, because I think science and technology has an increasing important role in society and how we deal with some of the societal challenges. But concomitant with the increasing role of science, there's an increase in resistance to science, and that resistance comes from fear and a lack of understanding. And I think that one of the ways to combat that is education, and I think there's no more powerful way than good scientific journalism. And so it's a pleasure to have Carl with us, and he'll be talking about the Genome Beat. Thank you for coming, Carl.
Thank you, Eddie. So I'm going to talk about what I like to call the Genome Beat, which basically means being a science writer who writes a fair amount about genomes. I actually started as a journalist, as Eddie mentioned, at Discover magazine, before actually science journalists really wrote about genomes. In the early 90s, certainly we were writing a lot about DNA, but we weren't writing about genomes. In fact, it was only a couple years into my work as a science writer that I started to realize just how important genomes were going to be, not just to science, but to my own life. And actually, the first moment that I really started to recognize just how important genes were going to be to science in general, just how much they could change the way that scientists view the world, had to do with dolphins. So I was working on an article about the evolution of dolphins and whales, and eventually this kind of expanded into become part of my first book, which was called At the Water's Edge. Now, up until this point, scientists who wanted to understand how whales and dolphins had evolved for the most part, they didn't look at genes. They couldn't. They would look at anatomy. And under the hood of a dolphin, it's just a mess. I mean, you think you're looking at a fish on the outside, but inside there are lungs, and in a female dolphin there's going to be a uterus, there are mammary glands. It's a huge brain in there. It's a mammal packed inside the body of a fish. Now, this is a very old problem. In fact, Linnaeus kind of struggled to figure out what to call them. Were they fish and were they whales? And then even in the mid-1800s, in Moby Dick, Ishmael is talking about whether whales are fish or mammals. And he ends this rumination with this passage. Be it known that, waiving all argument, I take the good old-fashioned ground that the whale is a fish, and call upon holy Jonah to back me. Melville is actually very interesting to read as an example of natural history right before Darwin. It's actually a wonderful book of natural history. I mean, I read it in college as an English major. These days, I read it as a science journalist, and I derive just as much pleasure from it. But along comes Darwin, and Darwin looks at this bizarre body of the whale, and he says, I think there's a pretty clear explanation for this. And in fact, it helped to support his theory of evolution insofar as he would argue that whales must have actually evolved from mammals on land. It makes sense that way. It makes sense that their ancestors had lungs that they were using to breathe on land, and they just adapted their anatomy to swimming around in the water. The question then became, well, okay, how does some land mammal get into the water? And on that score, it was a little tricky for him. He had been reading about some stories that a naturalist in the United States was writing about observing bears. And this naturalist had noticed once that a bear jumped in the water and was sort of swimming around with its mouth open. And Darwin said, hey, maybe some bear-like mammal a long time ago was swimming around with its mouth open and it would catch insects. And maybe that is how they started adapting to being in the water and they became whales. When The Origin of Species came out and this actual explanation was in there, that was actually something that Darwin's critics zeroed in on and mocked him mercilessly for. So much so that Darwin actually took it out of later editions. It takes a lot to make Darwin stand down, but this was one case where he figured that he didn't really know how whales had evolved. So the question had to wait. It had to wait for paleontologists to find intermediate fossils that would shed light on this. Some of the most important of these fossils were only first discovered in the early 1990s. This is part of what I was writing about. Hans Teveson at Northeastern Ohio Medical University found some fossils in Pakistan, and this is a relatively complete skeleton. This thing would have been maybe 6 or 7 feet long, and it had certain telltale anatomical clues that it was a whale. So, for example, in its ear bone, it has a thick inner wall called an involucrum, which only whales have. No other mammals have it. So this and other traits told Hans that we were dealing with a whale here. And this is what an intermediate whale, an early whale, would look like. A whale with legs, probably living kind of amphibiously, perhaps chasing after medium-sized fish, and then living a fair amount on land as well. So instead of the bear swimming around with its mouth open for insects, you have something that's a bit more maybe like, say, an alligator. And so Teveson and other paleontologists have been digging up all sorts of intermediate whales with legs that lived between, say, 50 and 40 million years ago. And these really do a wonderful job of starting to fill in just how whales evolved. But still there was the question, okay, if you go all the way back to when they were fully terrestrial, where did they come from? Where were those ancestors of whales? The paleontologists had their favorite. It was an extinct group of hoofed mammals called mesonychids. We think of hoofed mammals as being like horses or goats and so on. These are a bit more fierce. They had pretty big teeth, and some of them were predators. Some of them were probably scavengers. So in any case, so the paleontologists, they have their idea, mesonychids. So I'm working on this, I'm working on these articles, and then I'm working on this book. And you know, I've got the manuscript done. I'm commissioning an illustration, and this was the illustration that I showed, which shows all of these fossils and showing how the whales are closely related to mesonychids. This is all based on bones. Very late in the game. I mean, I'm dealing with like proofreading the book. I've been somewhat in touch with a couple molecular biologists who have been looking at whale DNA. And one of them says to me, you know, we got a paper coming out, and I think you might be interested in it. Now, these people were not looking at a genome. They were looking at maybe a few hundred bases of one gene. But they found something interesting. They were comparing whale DNA to mammal DNA, and the closest thing that they would find was to hippos, which is actually kind of a weird kind of thing to say, because if that were true, as in this kind of a tree, you have whales being closely related to hippos in a group called the artiodactyls. So this includes giraffes and cows and camels. This is not the kind of group that paleontologists wanted to put the whales into. This was wrong. I would talk to paleontologists and they'd say, no, trust us, this is not right. And you know, here was their logic. It's sort of an interesting kind of pre-genomic kind of scientific bit of history here. So artiodactyls, like cows and like hippos, they all have a very distinctive ankle bone. There is what they call a double pulley at the top of it, which sort of helps let the ankle move back and forth and lock into position. Artiodactyls have it, no other mammals have it. And mesonychids don't have it. Therefore, whales are not related to hippos. Forget about it. But, you know, I had a feeling that this was not something to be ignored, as some paleontologists would have it. And so in the book, I did say, well, okay, I'm going to show you this tree, but, you know, this could be wrong. and the geneticists might have something to tell us about this question. And I'm glad that I did, because the one little problem that the paleontologists had with their version of history was that they had not yet found a whale ankle. They had found lots of bones from whale legs and even, you know, whale digits and so on, whale toes. They had all of that, but ankles, for whatever reason, were kind of hard to come by. You know, you're grateful. I mean, you know, mammal paleontologists, they're grateful to find a tooth. They know an awful lot about mammal teeth, and they can tell you down to the species sometimes what you're looking at when you're just looking at a tooth. So, you know, they figured, well, eventually we're going to find an ankle. And they did. Around actually 2000, a couple years after my book came out, they started finding whale ankles. And those whale ankles, lo and behold, had that double pulley. So the geneticists were right. And so I wrote a non-majors textbook about biology called The Tangled Bank that came out in 2009. And science marches on. This is not the same tree as I had before. Now hippos have their happy place there. What's amazing to me is that, in hindsight, is just how little DNA it took to make such a big change to how scientists thought about a really interesting question. A question that kids ask about, a question that creationists use to challenge evolutionary biologists: where did whales come from? It only took a little bit of DNA to really just change the way people thought about it. But again, in the mid-90s, a little bit of DNA was all that scientists really had. And so we journalists were getting accustomed to writing about molecular biology and about genetics based on these little tiny scraps of information. Now, we knew that scientists had bigger things in store, and we started to follow scientists as they were not just trying to sequence, say, one gene. It used to be that a scientist might spend 10 years sequencing one gene. They were trying to sequence all the genes in something. And so I still remember when the Haemophilus influenzae genome was published. So this was in 1995, and it was, I have to say personally, it was kind of a mind-blowing experience. I thought, I'm looking at everything. I'm looking at the whole genetic landscape of this creature. 1,740 genes. I'm looking at what it takes to be something alive. And scientists have put that all together. Of course, the paper also included stuff like this, which is a little less mystical to look at. It's more like a catalog. And then there were charts like this. What does it mean for there to be a G plus C content of 38%? It was, while it's pretty mind-blowing simply that scientists could sequence a genome, what that told us was a bit less clear. So we reported on the publication of this first cellular genome, and it was important, but what it actually meant, we kind of skimped on a bit. And that kind of went for the genomes that came out after that. So it was quite impressive when animal genomes were sequenced. It was very impressive that scientists were able to sequence a mammal genome. That was great. And we knew that this was going to be important in the future, but what the genome itself at that moment told us was not quite as clear. And I think it kind of reached this kind of confusing sort of mix of excitement and catalog fatigue, reached its apex when Francis Collins and Craig Venter appeared with President Clinton to announce the sequencing of the human genome. And so you had this cover on Time magazine just as one example of the coverage that genomes were getting. I don't know if you can read it. It said, on the cover it says, "The inside story of how these bitter rivals mapped our DNA, the historic feat that changes medicine forever." Now, okay, that might be a bit of hyperbole, but it wasn't just hyperbole coming from the journalists. This was the kind of stuff we were hearing from the scientists themselves, and from President Clinton, and from everybody who was involved in sequencing the human genome. There was an awful lot of talking about what was going to be coming in the future. For example, how medicine was going to be changed forever. I mean, it's 12 years later, and I think we could fairly argue, when does forever begin in terms of applied medicine? I mean, we're starting to see a few cases where things are starting to translate into real real help. But the promises were so high then that that there has been a bit of a backlash. I'm not going to focus on that in my talk tonight because I could talk for hours about that. And it's a very important issue about what the human genome has done for medicine and what we should expect from it. But I want to focus instead on those genomes themselves. Because we're at this amazing point now where there are an awful lot of genomes that have been sequenced. Just today, I went on to JGI site, and I checked out the number of complete genome projects, 3,173. People will regularly sequence genomes to do just as part of some experiment. We have a lot of genomes, and they cover a huge range of species. And here are just a few: ticks, chickens, slime molds, cotton, mosquitoes. What I find a little puzzling is that an awful lot of them come with their own press release. This is a sort of a press release storing house, as a clearinghouse called Eureka Alert. So I just typed in the phrase genome sequence, and I got back 130 press releases. So we have press releases for the first lizard genome sequenced. We have a press release for the woodland strawberry genome. We have a press release for the first Irish genome, so that we can finally understand the Irish. It's a kind of a strange situation, and I've found it to be a little bit vexing. And I actually decided to diagnose myself, or to identify a disorder that journalists, some of us journalists suffer from, which is called yet another genome syndrome, which Jonathan Eisen, who's here, kindly abbreviated to YAGS for me. And I think that YAGS has a wonderful ring to it because it kind of expresses how I feel sometimes when I have to deal with the same old press machine banging away for another genome. It's like, another genome. I think I really developed a full-blown case of YAGS in 2010 with the sequencing of the zebra finch genome. Now, zebra finches are wonderful, and there's been a wonderful amount of research that has been done on them to help us learn about all sorts of things, including how new neurons develop. And I think it's a great thing that the zebra finch genome has been sequenced. But the press releases and the articles, the many articles that came out about it, were pretty much empty, I thought. So, for example, the articles kept saying that there are 800 genes involved in song learning in these birds. Like, that was like the hook. And I thought, well, what did you think? That there would be one? I mean, okay, so there are 800. Fine. What does that number mean? Something special to you? It doesn't mean anything special to me per se. So again, the articles all kept sort of looking to the future and kept promising things for the future, and the future will come, and we should report on things as they come. But at this point, it was just yet another genome with the same kind of somewhat meaningless reporting involved in them. So, I mean, I think that at this point, honestly, with genomes being, with so many genomes out, that it's a bit like someone looking through a telescope and then calling over to you and say, hey, look, I found a star. That's great, but there are a lot of stars in the sky, and you need to actually tell us something more about that star or about stars in general in order to be quite so excited. And so since 2010, when I diagnosed myself with YAGS, it has not gotten any better. And I just had a flare-up just in the past week or so. So the gorilla genome was sequenced. Again, wonderful, magnificent. I mean, gorillas are in our anthropocentrism, gorillas have to be one of the most interesting animals on Earth because they're so closely related to us. Gorillas are quite endangered, so this may help in terms of conservation biology. That's all fine. But it was just sort of painful to be looking at the press releases and press coverage of the gorilla genome publication and just not seeing much there. So here is one of the press releases that came out from the Sanger Center. Researchers announced today that they have completed the genome sequence for the gorilla, the last genus of the living great apes to have its genome decoded. While confirming that our closest relative is the chimpanzee, the team shows that much of the human genome more closely resembles the gorilla than it does the chimpanzee genome. So we have this story where, you know, we're being told a couple different things. One is that chimpanzees are our closest relative. Another is that gorillas have some genes that are more closely related to ours than our genes are related to chimpanzees. And then later it'll talk about how chimpanzees and gorillas have some genes that are closer than our versions. And this got lots of adulatory coverage. So here was an article in BBC's site. Gorilla genome could hold the key to the human condition. I don't even know what that means. But here's something interesting. If it really is such big news that it turns out that humans and gorillas have genes more closely related to each other than chimpanzees do, and nevertheless, chimpanzees and humans are the most closely related. If that was really news, then you wouldn't expect it to have appeared in, say, textbooks beforehand, because this is news. This is what the textbooks will later write about, right? So now that I've actually written a textbook, I can actually show you that that's not true. So this is my textbook from 2009, where I was writing about this phenomenon, incomplete lineage sorting, where some genes some alleles disappear from populations in such a way that, depending on the gene you look at, humans might look closer to gorillas or they might look closer to chimpanzees. Now, obviously, I was not talking about the whole gorilla genome comparison. And so this whole concept gets a much stronger footing now that there's a gorilla genome, a chimpanzee genome, and a human genome to look at. And that's great. But the problem is that in this urge to make the most of every genome that comes out, I think that the public ends up reading things in newspapers that does not reflect the reality in scientific research. The reality that this genome project adds a lot of insight to something that's been developed for a long time, incomplete lineage sorting. And this is actually this actually can have serious ramifications, and perhaps ramifications that scientists themselves sometimes don't appreciate. So what am I talking about? Well, here is a website from the Institute for Creation Research that just came out. The headline for this post is "Gorilla Genome is Bad News for Evolution." It says, evolutionists have long maintained that modern primate species, including, in their view, humans, are branches on an evolutionary tree that lead back to a common ancestor. True. But the recent news of the published genome sequence for the gorilla in the journal Nature adds more solid data to the growing problem facing the current model of primate evolution. And what they do there is they basically say, look, all of a sudden, we look at the gorilla genome and we see that humans are similar to gorillas in some cases and chimpanzees others, so therefore these evolutionary biologists don't know what they're talking about. And I think part of what they're doing is that they are playing on the perceived novelty of incomplete lineage sorting in this particular paper with the gorilla genome. So these things do have consequences, I would say. And I would also say that I'm not the only person who is now suffering from YAGS. I don't know how many people have it as well, but I do know at least one person, which is I blog at Discover Magazine, and my fellow blogger, Ed Young, I think he has it too. So Otzi is this hunter who died in the Austrian Alps some 5,400 years ago. His body was preserved and then discovered, I believe, about 15 years ago. And they've been studying him ever since. And just recently, I believe in the past few weeks, his genome was published. And so this is what Ed had to say. He said, after years of anticipation, the full genome of Otzi the Iceman, a 5,300-year-old mummy found in the Alps, has finally been published. The genome reveals Otzi carried a large genomic region known as the Y chromosome, which significantly increases the risk of traipsing around the arse end of nowhere with very little protective clothing and getting shot by arrows. As I recall, the big news in the Otzi genome was that I think that he had brown eyes. I mean, that's not important. And as Ed says at the bottom, as he added in a suffix, as Carl Zimmer notes, I'm suffering from YAGS. Now, okay, maybe I'm being a little tough, but none of this is to say that I don't write about genomes myself. I do, actually. In fact, these days, I think I'm writing about genomes a lot. I would say a fair portion of the stuff I write about involves genomes. And I just wanted to give you a few examples of the way that I'm writing about genomes now. So, for example, in Haiti in 2010, there was a horrible earthquake. The earthquake itself killed, I believe 200,000 people. Making matters even worse was that not long after the earthquake, there was a cholera epidemic. Haiti hadn't seen a case of cholera for 60 years, and yet suddenly there was cholera ravaging the country. I was just checking the statistics, and over 7,000 people have now died of cholera in Haiti. It's still going on right now. So there was an important question here. How did cholera get to Haiti? There is cholera in South America. There's cholera in lots of other parts of the world. This was actually an issue that triggered riots because a lot of Haitians were blaming UN peacekeepers for having brought cholera to Haiti. So how could this question be settled? I mean, how could you figure this out? Well, it turned out that by sequencing genomes, you could get to the bottom of this. And so this is from a paper that Paul Keim and his colleagues published last year in MBio, which I wrote about on my blog. And let's see, here we have the Haitian strains, and they are embedded in branches of cholera that come from one particular place. They come from Nepal. There were UN peacekeepers from Nepal in Haiti after the earthquake. Just getting this data was an amazing story, because the Nepalese government was not crazy about the idea of scientists coming in and possibly pinning the blame for this cholera outbreak in Haiti on their own country. And so Nepali researchers Nepalese researchers, they took great risks in getting this data from cholera strains in Nepal in order to show that, yes, indeed, it was from Nepal where this Haitian outbreak had come from. And this is, I think, a really important story, and it speaks to a story that we're going to see again and again in the future, in that when disasters occur, we have to be much more careful about the possibility of disease outbreaks adding to the suffering happening in these places. And sequencing genomes can be a way of either figuring out where these outbreaks come from, or one would hope being able to stop them from happening in the first place. Another genome story that I've just written about in the past year is the outbreak of E. coli in Germany. A pathogenic strain of E. coli started making a couple thousand people in Germany sick, and in many cases proved lethal. And so I got a call from an editor at Newsweek saying, "Could you write about this for us?" What was amazing was that, if I recall correctly, by the time I started sitting down to start calling people up to report the story, the genome of this strain had already been sequenced. They were using, I believe they were using one of these ion torrent machines that can do this in a couple hours. So we already I could already be talking to E. coli experts about the actual genome, the full genome of the thing that was killing off people in Germany. And that was important because by looking at the genome, scientists could see that this was a very strange E. coli strain. It was not the kind called O157H7, which you may have read about or work on yourself, that causes outbreaks in, say, hamburger, for example. It was a different strain that had not really been associated with so much fatality. And in fact, by looking at the whole genome, scientists could actually see how this strain had acquired lots of factors that made it so incredibly virulent. It was picking them up by essentially being infected with viruses and through other means of horizontal gene transfer. And this is actually something that more generally I think is incredibly important and only has come to light through genomes. And in my book Microcosm, which is all about E. coli and the history of the study of E. coli, I talk about how scientists, first they sequenced the E. coli K12 strain, this harmless strain that scientists use for lots of experiments. And then they started looking at these disease-causing strains. And the expectation was, well, we'll see, a basic E. coli genome that's been tweaked and adjusted a little by natural selection to turn it into a killer. And that's not at all what scientists found. So this was, again, when I think about sort of iconic images in my time in reporting on genomes, there's that Haemophilus influenzae image, that one circle, and then there were these three circles, this Venn diagram of genomes, where you have three different strains. You have, let me grab my thing. You have the harmless K12. You've got an E. coli that causes urinary tract infections, and then you have O157H7, which causes food poisoning. And only 39% of their genes are shared by all three strains. And there are some genes, hundreds of genes, shared by one strain and not by the other two, or shared by two and not by the other one. And what this speaks to is a really new way of thinking about evolution in general. So scientists have since then gone on and been sequencing more and more E. coli genomes. And I believe they're up to a couple hundred. I was talking with an expert who has been keeping on top of these. And so I asked him, well, how big is the core now, that central set of genes? And he said, well, around 3,000. But if you look at all strains of E. coli that they've sequenced so far in their genomes, this is from David Ussery at, I believe, University of Copenhagen. The pangenome is up to 50,000 genes, and it's still increasing. So the E. coli pangenome is much bigger than the human genome. And as I was saying before, this really is important. This really is what, as a journalist, I call news, because it changes the way we think about things. So this is an evolutionary tree of a number of lineages of bacteria and archaea, where they were just modeling it just based on vertical descent. So this is how Darwin would have thought about evolution. But these researchers also were able to identify lots of cases of horizontal gene transfer, and they tried to visualize it by showing lines going between the branches. This is more like what evolution looks like. The tree is still there, but it is just crisscrossed with strands. And for me, that's incredibly exciting, and that's something that you would not have found out without genomes. The last example I just want to talk about is something I wrote about for the New York Times about a little over a year ago, about a new branch of human evolution called the Denisovans. And without genomes, we wouldn't even know about this. So the story, some of you may be familiar with it, but I'll just refresh your memory and explain it to people who haven't heard about this. Svante Paabo at the Max Planck Institute for Paleoanthropology has really been a leader in reconstructing genomes from ancient DNA. He made big headlines for reconstructing the Neanderthal genome. And then the question became, well, maybe there are other ancient genomes out there that we should be looking for and reconstructing. So Paabo has created a big network of colleagues in many parts of the world, particularly paleoanthropologists who know where the bones are. And one of the places where the bones are is in Siberia. There's a cave called Denisova where there are abundant remains. There are lots of bones in there. Some of the bones are clearly human. Some of the bones are clearly Neanderthal. Some of the bones, it's kind of hard to tell. There are lots of mashed up bits of bone, little finger bones that you can't tell what they belong to. But this is a cave where humans and human-like relatives were occupying for tens of thousands of years. So there were some bones that were about 40,000 years old that turned out to contain some DNA. This is a picture of a tooth that had some mitochondrial DNA in it that turned out to be not human and not Neanderthal. It turned out to be something quite different. And then a finger bone, a little chip of a finger bone, turned out to have lots of DNA in it, enough to recreate the whole genome of something that is called the that they're calling the Denisovans. What's particularly remarkable about this is that it appears that the Denisovans have left some of their genome behind. This is from a paper from the American Journal of Human Genetics. Came out late last year. The dark parts of those circles are showing how many people in these populations are carrying some Denisovan DNA in their genomes. And so you can see that it's present in this area off the coast of Southeast Asia. It's not in Asia itself, which is very interesting because the Denisovans were found way up there. And so here's a variation on that kind of confusing tree that I showed you before. So before I was showing you a kind of confusing tree of bacterial evolution, which goes back billions of years. This is a confusing tree that goes back a couple 100,000 years. This is from the Reich paper, and so this up here would be the common ancestor of us Neanderthals and Denisovans. These are all humans. So you have a branch going down between, a splitting between humans and the ancestors of Neanderthals and Denisovans about 800,000 years ago. Denisovans and Neanderthals branch off about 600,000 years ago. And then you have these red lines, and those represent interbreeding. Humans come out of Africa, perhaps, there's a lot of debate, maybe 100,000 years ago, maybe less. It appears that they interbred with Neanderthals here and then spread out into Europe and Asia. And then as a wave of humans came down here, they interbred with Denisovans. And so clearly Denisovans must have had a huge range. Their range was bigger than the Neanderthal range. This was an incredibly successful lineage of humanity that then completely disappeared, although their DNA didn't disappear. And all that's left behind are a few enigmatic bones, a genome, and their presence in living humans. And this is going to be hugely important for many reasons, and one of which is that it's possible that Neanderthal genes or Denisovan genes that are present in us are going to potentially play some role in our health. Does having a particular Neanderthal gene make you more likely to get heart disease? Or, does it make you prefer the taste of woolly rhinoceros? Who knows? But at least some of these are going to be the questions that scientists are going to be asking now that they have these genomes. So, just to conclude, I would say that my experience in writing about genomes has firmly convinced me that we are in the middle of another scientific revolution, like the one in the 17th century, and that genomes are a big part of that. But it's important to focus on what makes that revolution so important. So in the 1600s, for example, one of the most important things that happened was that people invented microscopes. So I've been talking about E. coli and these other microbes, for example. Nobody would have even known they existed without the invention of microscopes. But again, it wasn't so much the microscopes themselves that mattered, but what people were seeing with them. Seeing the fine structure of an insect, seeing cells for the first time, seeing bacteria, what they were calling wee animalcules. Being able to see those things for the first time is what made the discovery of invention of microscopes so important. And so I think that if we keep that in mind, then I think we can, once and for all, find a cure for YAGS. So, thank you very much.
Questions for Carl?
There's someone right there.
Hi. I couldn't tell whether in your YAGS discussion you were sort of complaining about the people who are sequencing genomes and publishing them, us, or if you were complaining about your scientific journalism colleagues who are often, is this the key? Is this the thing we should be writing about? And no matter how much nuance you try to have in those discussions, when it comes out in the press release, it's often the key to, gorillas are the most important thing this week. So how do you, what should we do about this? How should we cure YAGS from the perspective of the people who are doing the science?
That's a very good question, and I do not mean to. I
No
hope,
offense taken.
Oh, good. I do not. I certainly do not mean to say that scientists need to stop sequencing genomes. That is not what I'm trying to get at. You know, it's just I have this sort of reaction when I'll tell you when I have this reaction when I read articles that claim that, you know, gorilla genomes hold the key to the human condition, whatever that means. I react this way when I get a press release saying, you know, we've sequenced the first Irish genome. And I also get this when I get a call or an email from a scientist saying, oh, this is so exciting. This is big. You know, we have, for example, the first lizard genome. And I have had all those experiences. And so I think that all these things end up giving the impression that the sequencing of a particular genome, in addition to being a huge accomplishment, which it is also a kind of a game-changing, headline-making story that changes the way that we see things. And the fact is, if you look through these press releases, and you know, and many journalists do, they then try to, you know, they then make a story out of it where there's not much there. That's all I'm saying. There are lots of cases where sequencing a genome tells us something important. That's when I want to write the story. That's when I call up my editor and say, I want this one. You know, I'm not going to call up my editor and say, oh my God, you know, the strawberry genome has been sequenced. You've got to give this to me. You know, and you have to bear in mind that, you know, I'm just talking specifically about, say, newspapers and magazines. It's not like there's an infinite bandwidth for stories. So reporters and editors need to decide, they need to make judicious choices about what's going to go into say, a science section, like the Science Times. Some of the stories are just going to be fun, and I like to write fun stories myself. A lot of them are going to be, you know, this is newsworthy, this is important. They're going to only be a tiny subsection of all the science that is done. You know, every week we get hundreds upon hundreds of press releases that people have decided are so important that we need to know about them, and we can't write about them all. And I just think that I just find that there are too many stories and press releases about the next genome. That's the point that I'm making. So I mean, you know, science is a funny business where it's very hard to tell a scientist or a public information officer, you know, maybe you don't need to write a press release. You know, that doesn't make it bad science, but you know, maybe this doesn't need to be written about.
Over on your right, Carl.
Yes,
you have many survey or overview of many different scientific cultures in different areas, and they have different levels to which a statement is accurate. So there's precisions of measurements are different in chemistry and biology and physics. In principle, genome sequencing is natural product structure elucidation, right? It's telling you where the carbon atoms are, the hydrogen atoms, the oxygen, the phosphorus, and so on. Atoms are in a particular set of molecules that happen to be involved in inheritance. But unlike natural structure product elucidation from the German 19th century, the level of precision in any genome sequence is far below what any of them would have accepted as a structure proof. But I guess the question I was going to ask you is whether you have an overview or an opinion of this, because you look at biology and they look at astragalus, and obviously there's the double pulley bone is, you know, imprecisely measured by the chemical standard as well. And do you think that the imprecision in genome sequences has anything to do with why they're not so widely used in medicine so far?
Why they're not used in medicine so far.
Well, why they haven't revolutionized the future of medicine forever.
So, I mean, it's been fascinating to me, sort of watching the study of genomes mature in terms of medicine, but with maturity comes humility, I think. I mean, in the sense that, I think for me, what makes the strongest impression are things like these genome-wide association studies, where any particular gene gets associated with a tiny, tiny, tiny bit of variation. And so that it becomes, so that you can't sort of zero in on a particular gene and say, well, if we address this particular gene, we'll be able to cure heart disease or cancer or so on. What scientists are telling me is that what it's helping them get at is the pathways that are involved in these diseases. But again, that's going to take a lot. It's going to take time to work through those to get to something. I mean, I guess I'm not sure that the imprecision that you're talking about has been an issue so much that I've seen. But maybe there's something that I haven't observed yet. I mean, I guess that these GWAS studies have been so high profile that they're the things that have made an impression on me in terms of the role of genomes in medicine.
Thanks, Carl, for your presentation. Increasingly, the scientific community is being asked by funding agencies to include outreach and public education components in their work. Here's the question for you: how can scientists do this and still do their work, obviously? And secondly, we seem to be dealing increasingly with a public and politicians that are very skeptical of scientists and actually throw red herrings up in the air constantly. And I'm thinking like global climate change, and my colleagues at the University of Washington are dealing with this. And obviously, the scientific evidence is overwhelmingly demonstrating that this is indeed something that's happening, but still the public remains very skeptical, and politicians constantly say, additional studies need to be taken in, so we can really see that there's global climate change. And yet the scientific is so frustrating for scientists to try to communicate their science, but I think it's also a responsibility of scientists that are being funded by the public through U.S. tax dollars to do this. So how can they best do that, and how can they sort of deal with the skepticism and the doubters that are out there that are kind of seem to get the headlines constantly?
So, certainly the first step is to go beyond just sitting in the coffee room with other scientists complaining about those people who don't appreciate the science. I mean, scientists, I find, are very good at, grousing about it and then expecting something else to happen. I don't see why you'd make that assumption. Part of the problem, I think, is that often when scientists do try to communicate, to reach out, they don't really know how to do it. That comes down in large part, I think, to the fact that as scientists are educated and trained, there's so much focus put on doing the experiment right and so on, that no one actually gets any training really at all at, for example, writing or speaking clearly. And so there's an attitude that that'll just sort of come along. You sort of pick it up on your own. It's not an important skill. Which is interesting to me because scientists, aside from writers, the people I know who write the most are scientists. Whether it's writing up experiments or writing grants and writing lectures, for example, and yet there's very little focus put on that. I teach a workshop at Yale for science graduate students there. And I do a fairly cruel exercise. I pick someone at random and I say, just tell me what you do. And just you can't assume that I'm, know all of the jargon involved in your field, but what do you do? And it's very difficult for these graduate students to say that clearly and compellingly. And so they'll try to say it, and I'll stop them, and it's like, no, try again, and we'll go through it. It can take 5 or 10 minutes for them to say what they do and to give a sense of why it's important to them. I mean, we're not talking here about whether you can convince a global warming, quote-unquote, skeptic that global warming is real. Here we're talking about something even more basic. What is science? What does a scientist do? Why does a scientist, spend 15 hours a day studying something? It's got to be important, right? And yet, scientists have very little practice at articulating these things. And it takes work. So I know that scientists are busy, but I think this is just an important part of training for scientists, because scientists are not simply robots at a lab bench. Scientists are part of our society, and being part of that society means being able to talk, to write, to communicate to other people, to share the insights of science. Yes, sir.
I want to point back to the slide you showed where you showed the guy looking through the telescope up at the stars and said, okay, yet another star, right? Right. And for diagnosing something that I think a lot of us felt but didn't know what the technical term was on YAGS. I think one of the with the astronomy parallel, one of the parallels with genomics is the gigantic deluge of data that astronomers deal with and that we deal with. Yeah. And astronomers have sort of reached out to the public in a lot of ways. These data are available, just like genomic data are available to the public. But the public has really embraced, at least many highly trained amateurs have embraced a lot of that data analysis coming out of astronomy and related fields.
Yes.
I think back to the origins of like the SETI at home sort of thing. Mm-hmm. But no similar culture that I've seen yet has sort of developed around analyzing genomic data among talented amateurs. We've seen a little bit about people kind of creating labs in their own homes. But I'm just wondering, in your travels, have you seen any sort of subculture like that starting to develop in the same way that people, search for, new things in the sky using the publicly available data?
Right. Yeah. So the question is about what might be called citizen science, and it is true that in astronomy there are many initiatives that bring together amateurs and professionals. And so, there's long been a tradition of amateur astronomers just using their telescopes and finding new things, discovering supernovae, for example. More recently, there has been, I think it's called Galaxy Zoo, where it's a website where people will just go in and they just classify galaxies for fun. And there are thousands of people who consider this fun. And actually, they've been discovering new properties of galaxy formation through this crowdsourced effort. But this is not limited to astronomy. And so in biology, for example, some of you may know about Foldit, which is a protein folding game. Basically, it started out as just a computer program that would try out different configurations of proteins, and the people who were running the program asked people if they could just hijack their computer and use it while it was sleeping, and people said, sure. And as a screensaver, they would actually show what the computer was trying. And they got an email once from somebody who said, is there any way that I could just kind of move that protein over a little bit? Because I can see where it should fold, and the computer can't find it. It's driving me nuts. And the scientist was like, oh, yeah, hold on a second. And they went off, and they wrote a game. They gamified this so that you could score points and for the number of things you had folded, and there would be this sort of communal effort. And so now Foldit is this huge burgeoning community that is finding out important things about the way proteins fold. And so I believe it was last year, the folded community published their first paper in Nature, and it was amazing. You look at the title, and then below it, there are a couple names of the leaders of the scientists who started the folded program, David Baker and a couple other people, comma, and 57,000 co-authors. That's how many people were involved. And I believe that there's actually something similar happening in evolutionary biology and bioinformatics. I think it's called like Philo. Does anyone know? Can anyone help me with this? Yes? Philo? Okay. Ends in McGill, I think. I'll put it on my blog, okay? Tonight I will just post it on my blog just so that people can see it. And basically what the game is that you try to match just homologous stretches of DNA in different species. And there are people who find this fun, and they're actually getting data out of this. And so I think this is a huge opportunity to do good science, but also to then, getting to this issue about reaching out to the public and making people understand about science, that it's not all just settled and there's nothing new to discover, that you yourself as an individual can be part of the discovery. I think it's happening quite widely in other branches of biology, more kind of natural history. For example, there's a big firefly project in the eastern United States out of the Museum of Science in Boston, where they're trying to chart firefly populations and getting data they could not get on their own. And the internet makes it possible to gather these things. So yeah, I think that genome people should be thinking about this. The key, I think, is to make the interface just addictive. And then people will show up. We already know that they are in other cases. And you guys are good at computer interfaces, so, you can do this.
Okay, the last question is.
Hi. So you talk about having YAGs, and
Yes.
so one thing that we've been trying to do is annotate genomes computationally, and I just wanted to hear your perspective on, when did that really start? When did we really start annotating genomes computationally? And also the second thing that I wanted your perspective on is when I go outside of academia and I say I'm a bioinformatician, they say bioinf for what?
Yep.
And hidden in everything, all the studies you're talking about are a lot of algorithms, a lot of computational work. And I'm wondering, why does bioinformatics get lost in your opinion when it's such an important part of teasing out the biological insights that we want from the data?
Yeah. I don't know. That's a good question. It took me a while as a science writer to come across the phrase bioinformatics. I don't know when the word was originally coined, but I in the business of writing about this stuff, didn't come across it for a number of years. I think that bioinformatics is so much sort of in the service of other questions that people don't think of it on its own. It's not a great sounding word either, but that's just more kind of marketing. And what do you call it when you do bioinformatics? A bioinformatician or something? It doesn't roll off the tongue. But and how you would bring bioinformatics more into people's awareness is definitely a challenge, because it's one of these kinds of science which is really important, but when it comes time, say for example, for me to write about it, it's tricky because you're not a biologist in the Australian outback stalking the world's most venomous snake. You are at your computer, coming up with an algorithm. But it might be possible. Maybe gamifying bioinformatics will do for bioinformatics what journalism has yet to do. Perhaps. I don't know. Anyway.
Okay, well, I'd like to thank you, Carl. That was
Thank
terrific.
you.